Hello, everyone, and welcome to Arthritis at Home. My name is Ellen, and I'm the programs coordinator. Today, I'm joined by Dr. Simon Wisnovsky, an assistant professor in the Department of Pharmaceutical Sciences at the University of British Columbia. Very recently, himself and his team were awarded the Ignite Innovation Grant from the Arthritis Society for their project titled Characterizing Aberrant Autoantibody Glycosylation as a Driver of Inflammatory Arthritis Pathogenesis. I know that sounds like a lot, but don't worry, we're going to distill it down. Very, very exciting work. Dr. Wisnovsky, thank you so much for joining us on our program today.
Thanks so much. Glad to be here.
And I ask, do you mind telling us a little bit more about yourself and how you're involved in rheumatology?
Yeah, of course. So I recently started my research lab at UBC, and just from a kind of very broad, like 10,000-foot view, my lab focuses on a field of research called glycoimmunology. And what that is essentially is studying the way that sugar molecules that cover our cells and which cover a lot of the proteins that our cells produce, how those sugar molecules affect and regulate the activity of our immune systems. And so that's kind of the key connection there to rheumatology and to arthritis. Is it turns out, and we may talk a bit more about this later, that changes in we call this glycosylation. I'm kind of falling into that, but essentially glycosylation is just a kind of fancy word for saying, the attachment of sugars to proteins. That changes and kind of alterations in glycosylation and in the chemical structure of these sugar molecules are actually some of the very powerful predictors of whether a patient will or won't eventually develop inflammatory diseases like arthritis. And so this is a kind of really exciting new direction for my lab, is kind of trying to harness this kind of general knowledge we have of glycoimmunology and try to really apply that to this kind of really specific disease context of rheumatology and arthritis.
Incredibly cutting edge. And I just, from a patient perspective, I think, thinking about what we know, right, about immunology and understanding that inflammatory arthritis is an autoimmune, right? Our cells in our body attack the rest of our body. Just think to ourselves, wow, like we really don't know that much about these pathways, and that there are leading scientists, dedicating their time or their lives to examine the smallest little thing that could lead to innovation. I'm, very, very intrigued by your work. And could you tell us a little bit about autoimmunity and what drives autoimmunity? Maybe the role of, like you said, those sugars, or sometimes known as glucose.
Absolutely, yeah. So, I mean, the way I would frame it just to start with is, our immune system operates in this very finely tuned balance, right? Whereby you want your immune system plays this incredibly important role in kind of keeping you alive and healthy and kind of walking around in the world. And obviously, you want your immune system, as part of its function, to be able to detect when, say, you're infected with a virus like SARS-CoV-2, or you have a bacterial infection. And you want your immune system to be able to really effectively activate and initiate a kind of powerful response that destroys those types of infectious pathogens. On the other hand, obviously, if your immune system gets kind of too amped up and too hyperactivated, you can end up with a situation where the immune system can incorrectly attack healthy tissues, or incorrectly attack your own body. And so this is, I mean, I think you alluded to this, and I think it's kind of the key concept of immunology to understand, is just because you need to kind of finely balance those kind of two considerations. So having enough immune activation to effectively destroy an infectious disease, but not so much that you have kind of autoimmunity or the immune system attacking your own physiological tissues. It makes immunology incredibly complicated, because you need you need to regulate the activity of your immune system at so many different points, and have all of these different kind of checkpoints and feedback loops that allow you to kind of control and modulate and kind of tune the activity to exactly the right level. And so I think what happens in autoimmunity essentially is that something happens, and that something could be a lot of different things, to kind of tip that balance out of control. So basically, you stimulate the immune system such that it's kind of hyperactive and attacking tissues that it really shouldn't be attacking, that it should recognize as kind of self-healthy tissues. And what drives that in inflammatory, and so that is the disease of arthritis, essentially. It's an autoimmune disease where we're having inappropriate activation of the immune system. And what drives that, I think, is actually a really fascinating ongoing field of research. And I think in terms of developing treatments for arthritis, that's kind of the key question, right? Is what's causing this and how can you kind of target it with a drug? I think specifically one of the areas that my lab studies are that, I'm sure people on this listening to this will have heard of antibodies, which are the immune system produces to fight infection. One of the things you find in a lot of forms of arthritis is that your immune system actually produces antibodies that bind to your own tissues. And we're interested in kind of defining, again, those like really kind of detailed layers of regulation by which, how do those antibodies kind of play a role in kind of activating the kind of downstream amplification of an immune response, of an autoimmune response in arthritis.
Absolutely. And, thinking of these mechanisms and these very intricate pathways, specifically your lab is looking at, you had alluded to these carbohydrates or these sugars. Can you tell us what role does sugar play? Is it the same as dietary sugar?
Yeah, that's a good question, actually. So basically what happens is that your body, these sugar molecules are not quite the same as the sugars that you eat, but what happens is that your body will eat glucose, eat fructose, these things that, these kind of common terms that you all have heard of in your food. But then through a kind of complex metabolic process, it'll convert those sugars into all different types of other kind of chemically similar but different sugar molecules. So these are probably things that you haven't heard of. So like sialic acid or fucose or N-acetylgalactosamine. These are kind of chemical derivatives of the glucose molecule. And your body essentially uses these sugars to essentially like tag proteins that are expressed on the surface of all of your cells. So obviously all of our cells are made up of protein, but those proteins are kind of decorated with this incredibly complex collection of sugar molecules. And one of the roles this kind of plays in the immune system is that immune cells actually kind of float around the body tasting these sugars. And they do that through these kind of complex binding of what are called receptors to sugar molecules. And so different receptors will bind to specific chemical structures, different types of sugars. And some of those receptors will kind of, again, tip that balance towards immune inhibition or kind of reduce immune activation. And some of those receptors will enhance immune activation. So for example, we have receptors on our immune cells that bind to sugar molecules that are only expressed on fungi or on yeast. And that's a kind of signal that tells our body, okay, we have a yeast infection, we have to activate, and kind of try to destroy this pathogen. And then similarly, we have immune receptors that bind to molecules that are only expressed on healthy cells, on our normal cells. And those receptors tend to tamp down immune activation, so to kind of reduce the immune response because it's a kind of signal of self. So basically, these immune cells through this incredibly complex biochemical interactions kind of have a way of kind of saying like, oh, this cell tastes bad to me, I'm going to try to activate and try to destroy it. This cell tastes good to me, has the right type of sugar, I'm going to ignore it. And so that is kind of the way in which these sugars kind of play this kind of really central role in kind of maintaining that balance I spoke about, that kind of immune balance. Where I think this plays into arthritis is that there's a lot of interesting research that's come out now from other labs that's shown that these antibodies in particular, but also other proteins that are kind of expressed during arthritis, have weird sugars attached to them. So they have sugars attached in kind of the wrong place, or the sugar is the wrong structure. And that is a kind of characteristic of antibodies that you see in arthritis patients that you don't see in normal patients. And so the question is that we're interested in asking is like, what is that doing in arthritis pathogenesis? So it's like pretty well established fact that this happens, but is that, for example, causing an antibody to bind to some receptor that's then activating an immune response? And so that's kind of what the question we're interested in answering as a lab.
Amazing. And let me know if I'm getting this correct. So we eat sugars, so to say,
Mhm.
right? Different kinds of sugars, whether that's a complex carbohydrate in the form of like whole wheat bread or an apple that has a little bit of fructose, a little bit of glucose. Our body then uses it, right? We use it to create things. And some of these things are signaling molecules. So you take the sugar, you kind of rejig it. So you take your Lego, you put a
Yeah.
few things into different spots, you get a new fancy name, and they act as, they act as different things in our body.
Mhm.
In a perfect world, that would all be good. Everyone is happy. We're eating something, we're then transforming into something our body can use. You're saying in inflammatory arthritis, what happens is the rejigging process, the putting the Legos in the spots, that process creates a molecule that doesn't quite look like something we can use in our body or has a weird funky shape. And that is specific to individuals with inflammatory arthritis. And now we're trying to understand why is the Legos, why are they being put in slightly different places? Why is that sugar molecule not, first thing, doesn't look like, and then doesn't act like, and doesn't play the role that it's supposed to play? Is that correct?
That's you nailed it. I think that's exactly what's going on. And I really like the Lego analogy actually, because when I mean, in my kind of field, the way we kind of represent these sugar structures visually is through these kind of they're almost like these cartoons of kind of these square boxes or blue circles or things like that to represent each specific sugar kind of attached together in chains. And they actually do kind of look a bit like Legos when you kind of when you visualize them like that. So I think that's a really good analogy.
Awesome. So can I ask, what do you hope to understand through this kind of like, base level project, right? Where you're really just gathering information. And how do we hope to then translate it into maybe like a second project, or what does it mean for patients?
Yeah, so I mean, this is, as I said, a new project for us, and I think we're very much at the kind of exploratory stage. But that being said, I mean, I think what we're really hoping to identify here is to figure out exactly how that changed glycosylation that I talked about. How does that affect the interaction of these molecules with different immune receptors? And so the hypothesis that we're posing is that when you have this kind of new form of glycosylation, that might shift again. I keep coming back to this concept of balance because it's really important, but that might shift the balance in terms of how the antibody is interacting with kind of inhibitory immune dampening receptors versus activating immune stimulatory receptors. And maybe that is kind of one of the key things that's going wrong in arthritis is when you get this kind of introduction of these new types of sugar molecules, it kind of leads to this potentially this initial kind of disruption of the balance and a change in the signal that then initiates all these kind of downstream inflammatory effects that we see in arthritis. And so the dream of our research is kind of if we can kind of identify exactly what those receptors are, those specific proteins are that are binding differently to these autoantibodies, you could design a drug to block that. And that's kind of the key, and again, that's, years in the future, but, I think that's ultimately the goal of our research is to try to identify these interactions that are regulating the immune response. And then once you kind of understand those interactions, that gives you the ability to really design drugs in a kind of rational way such that you can block or modulate the interactions for some benefit in the long term for patients, for some therapeutic benefit. And that honestly is, I mean, that's kind of my whole pitch as well for like basic immunology research, because, so many of the drugs we have right now that, cancer immunotherapies, but also immunotherapies for arthritis and for other diseases, they really come out of studying the kind of nitty-gritty details of these specific interactions that are kind of tuning this balance of the immune response, and then identifying, well, what is, if we want to make a drug, what is the specific receptor or the specific signaling molecule that we really want to target? And so that's kind of how I would see our research kind of fitting in there.
Absolutely. And I was actually going to reference cancer as well as diabetes because I know they have looked at similar pathways. And as a patient, I'm excited and so looking forward to the fact that now you're doing this in arthritis, right? To be able to look at specifically what's going on. And for one watching, right, it's truly through the study of something so small that we're able to then, test and test and test study after study after study through these different phases to then create, right, what we have on the market today as available drugs. So I know this seems very far off than you taking, a medication, but believe me, this is step one. So with that, I do want to thank you for your time, Dr. Wisnovsky, and the incredible work that yourself and your lab are doing. Do you want to take any time to give a shout out to any members of your lab or the Arthritis Society?
Absolutely, yeah. I mean, I'll shout out. So Natalie Stewart in my lab is a recently joined PhD student who's going to be starting work on this project, and she's amazing. And so I know she's excited to start work on this. I also just want to say such a big thank you to the Arthritis Society for this support. I'll say myself. I mean, I think one of the, I think this having these kind of Ignite fellowship grants, I think, from the Arthritis Society is so valuable, because I think as as someone starting running their lab, I think one of the hardest things to do is to kind of get a little bit of funding to kind of test a really new idea. And I think that that's such an important thing to support in terms of research, is kind of supporting labs to kind of really try to advance new ideas and new things. And that's a kind of something that the Ignite mechanism is really focused on. And I think that's such a terrific thing. And we're so kind of grateful and thankful for that support from the Arthritis Society.
Absolutely. And to any viewers who are tuning in right now and you perhaps do support the Arthritis Society, just know that, your support of the Arthritis Society is allowing researchers to do this work. So thank you as well. With that, I'm going to say, hope to see everyone again on our program. And we're going to thank again, Dr. Wisnovsky, for your time, your energy, your expertise, and all the work that you do.
Thanks again so much.
Take care. Goodbye, everyone.